Okay, so now I'm really excited to be able to introduce Kayla Rinna, who's a graduate student at Eastern. And Kayla actually worked with us a number of years ago after she finished her master's. She was with us for a year. And the work that she's going to tell you about, she actually was the brains behind it. So we had an interesting case that you'll hear about that then Kayla really went into the literature, came up with some innovative approaches. And then we were able to build on that in her absence. And we've, again, gotten our claws into her and brought her in. So that's a consistent theme that we like to try and drag people in. So Kayla.
Thank you. So good morning, everyone. Thank you so much, Dr. Hempstead, for the introduction. He actually kind of just walked you through how I was going to explain to you all how I came up here. So again, I worked at RPCBI back in 2019. I left in the fall of 2020 to pursue my doctorate degree in clinical psychology at Eastern Michigan University. And so, like he said, I came back last fall as a research supervisor as I finished up my doctoral training at Eastern. And so I'm excited today because, again, I'll get to revisit a lot of the work that I did previously and then talk about where the team was able to take this project since I left. And so it's been really fun, very full circle moment for me. So to start off, no conflicts of interest, but I do want to call attention to some of the grants that are ongoing and have funded these projects. And so for this presentation, my overall goal is showing the use of multiple characterization methods and translating those methods into treatment plans at the individual level. And I'll be using individuals with a diagnosis of posterior cortical atrophy as an example. So to begin, what is posterior cortical atrophy? So posterior cortical atrophy, or PCA, which I will be referring to that throughout the rest of the presentation, is a relatively rare neurodegenerative disease, often caused by the atypical accumulation of Alzheimer's disease pathology in the occipito-parietal and the occipitotemporal areas. So as you can see, this image, if I can get the mouse to work over here, this image is from an article from Brain in 2016, and it shows a distribution of tau. And so it highlights those areas that are affected. This syndrome is characterized by visual perceptual and visual spatial impairment within the context of relatively preserved visual acuity. So folks with this diagnosis have quite a long process to get diagnosed, often thinking that it's first eyesight that's going, but really it's an underlying neurodegenerative process that's happening. So for example, color and depth perception may be affected. So someone with PCA might see this image and think that there's a step there, or not be able to tell if there's a step. Additionally, another common symptom is that individuals might be able to see objects on their own. However, when they're presented side by side, multiple images on a screen, or if they're overlapping, they may no longer be able to distinguish between them. And so identifying and distinguishing between various neurocognitive disorders is crucial for providers, patients, and caregivers, because it allows us to understand disease progression and identify intervention points. So individuals undergo multiple forms of assessments to capture their diagnosis, including neuropsychological testing and imaging such as positron emission tomography, or PET, and magnetic resonance imaging, or MRI. So PET scans, specifically FDG PET, identify patterns of regional glucose metabolism in the brain and are an effective method for differential diagnosis of Alzheimer's pathology, such as PCA. So if you see the image over here, you can see down in this left side, it demonstrates some hypometabolism. And then MRI provides detailed images of the brain anatomy, vasculature, and functional parameters like blood flow. So for example, arterial spin labeling, or ASL for short, is a technique that measures blood perfusion, which reveals reduced blood flow patterns in the affected areas of Alzheimer's disease. So again, the image on the right, if you see the bottom half, it's darker, that's showing less cerebral blood flow in the bilateral areas. So while imaging methods are instrumental in comprehending symptom presentation and disease progression, it's crucial to grasp how these affected areas are influencing an individual's daily life. So this is particularly pertinent for folks with PCA, where eye movement plays a pivotal role in visual perception, potentially impacting various aspects of daily functioning. So our team, to better try to capture these eye movements, we developed a simple eye tracking task. So this task consisted of just basic black and white line drawings on a white background. And these images were separate, and then they eventually moved closer together until they were overlapping. And so we used this to paint this as an example. This is someone's baseline testing. And you can see that the number of fixations across images demonstrate some variability and quite a few off-target fixation points as well. Which now brings me to my first case study, which this is pulled from. So our first case study was a 77-year-old right-handed African-American male with a diagnosis of PCA, and he came to us in 2019. His symptom onset was about 2 years prior, with difficulty with visuospatial processing and functional abilities like reading text. So again, at the time, we did not have PET data. So as you may recall, we visited the literature to find areas that might be affected by PCA. So we then developed an individualized, so sorry about that, montage that targeted the bilateral occipital region. And this was for TDCS. Now, this participant completed a baseline neuropsychological testing, eye tracking, and fMRI, followed by 30 sessions of TDCS. Following these sessions, we then repeated fMRI, eye tracking tests at day 30, so their last day of their TDCS session, 24 hours post their TDCS session, and then again later, a few months later, as follow-up. So a few slides ago, I showed you their baseline testing, and now this shows the eye tracking example post TDCS. So just to orient you all, the blue shows their baseline fixation points, and the orange shows their post TDCS fixation points. So as you can see, just by visually inspecting this, the post TDCS fixation points are much more on target of the areas of interest. And as a reminder, we targeted the primary occipital cortex, and we saw an increase in occipital activation when we evaluated change from baseline to post using an fMRI version of the eye tracking task. So as you can see, it's activated during that task. And using the Con toolbox, we saw an increase in the magnitude and extent of the visual network connectivity. And these complement the eye tracking data and demonstrate a physiological response in brain regions that mediate visual processing. Now, after piloting these methods with case study 1, the program expanded on this project and began recruiting folks with PCA specifically. So this pilot study was funded by Dr. Hampstead's R35 and University of Michigan discretionary accounts. After screening for eligibility criteria, the final sample of this pilot was 12 individuals with PCA. So everyone enrolled in this project received a baseline MRI, neuropsychological testing, eye tracking, and PET. So they received FDG, amyloid, and tau. And to personalize areas to target for stimulation, we used FDG PET results to identify areas with primary hypometabolism. So as you can tell by these images, I'll point over here for you all. Areas that have lower hypometabolism from the FDG PET were targeted for stimulation. So some folks received left stimulation, bilateral, or right. Participants then received 20 to 30 sessions of active TDCS concurrently with a visual task, and stimulation lasted for about 30 minutes. After treatment sessions, individuals completed a repeated MRI and testing the same day as their last TDCS session, 24 hours post TDCS, and again 30 days later. So that simple eye tracking task that we used before, we used it again. However, when I left, the team began getting cruel and made these tasks a little bit more complex. So as you can see, the new task, the complex task, has a colorful and distracting background. So the results with these folks show that compared to the simple eye tracking tasks, participants do relatively well, except for the separate complex condition, which is the example that I have up here in the bottom left. However, if we look at their results from baseline and then look at the change scores from post TDCS and then 24 hours TDCS, we actually see that they do show some improvement. So then our question is, what might be driving these improvements? We know that the networks, so we know that network segregation, or how independent these networks are from one another, declines in normal aging, but it's further affected by neurocognitive disorders like PCA. The team then decided to look into functional connectivity analysis and compared the PCA participants with 37 healthy controls. And as you can see, the participants tended to show improvement in the visual network after TDCS relative to before. And then we also know that the dorsal attention network is a top-down control, which also might be affected by PCA. And we see the same trend where individuals show a general increase from pre to post intervention, which aligns well with the healthy controls. And then, so taken together, this shows some physiological evidence that maps onto the eye tracking performance. So we also wanted to examine if stimulation is changing the rate of blood flow in the brain, both globally across gray matter, as well as within specific functional networks. Where the entire group showed an increase in functional network segregation, as shown on the previous slide, only 2 people showed consistent increase in cerebral blood flow. And this increase was observed globally as well as within the visual network and dorsal and ventral attention networks. So then our question is, what about these 2 individuals are causing greater response compared to others? So we decided to look into those 2 individuals, who I will refer to as BBC9 and BBC12. So first we decided to look into their baseline to see how do they perform on their neuropsychological testing. And we didn't see any differences from the rest of the cohort. So then we looked into their eye tracking, and both showed increase from baseline to their last day of TDCS, and then a slight dip 24 hours later. But if we look at the results from the eye tracking, they're still higher from their baseline. So overall, demonstrating some improvement post-treatment. So and then we asked again, we got more curious and wanted to review some other physiological changes that might be going on with these 2 participants. When we looked at their FDG PET metabolism, particularly FDG, and ASL complement each other. So we were just wondering, maybe there was something going on with that. And again, they don't really stand out from the rest of the cohort. So then we dug deeper and we looked at their amyloid and tau. And as you can see, so for BBC9, if you see the blue and the yellow dots, yellow corresponding with BBC9, there's really no consistency between amyloid and tau except for the dorsal attention network. There's slightly higher amyloid there. However, for BBC12, we can see consistency, higher amyloid and tau across networks. So what does this mean? We don't know yet. There's not a very consistent story here, but we are trying just to see what about these 2 folks might be standing out. So bringing it back together and trying to take this individualized approach, we demonstrated using multiple characterization methods and then translating those methods into treatment plans at the individual level. And when we did this at the individual level, we did show some evidence of cognitive and neurophysiological changes via eye tracking and MRI across the group level. And we're also able to see that at least 2 of these individuals had an increase in cerebral blood flow. So what next? I think natural what next questions are increasing the number of people that we can recruit for our cohorts, perhaps looking at other presentations of Alzheimer's pathology and comparing, seeing if we have any patterns that strike us or stand out. And then if I were to put my clinical psychology hat on here, I think something to also consider. So we do see some sort of change pre to post TDCS. Now, how is this mapping onto their everyday life? So I think getting some ecological data, asking how folks are engaging in their everyday life post TDCS, see those changes and how that might map onto these cognitive and physiological changes as well would be a great idea. Thank you for the team. Thank you for everyone to come here and listen to me. Any questions?
Any questions for Kayla? Yeah, just
yeah. Yes, please do.
Yeah, very nice work. So we have results of the GAN. You guys also looked at default mode network. I think I saw some hints in the beginning data that there may be, but anyway, you tell me if there's anything meaningful.
We did, and Dr. Ham said it actually would have a better response to those results. I don't want to assume here, so I'm going to let you.
Yeah, so we did look at, Alex did segregate all the networks, and I don't recall the functional connectivity. I think that we did with that particular group, we did see the biggest effects in the dorsal attention and the visual network. Other networks were a little bit all over the place. But I think one of the things that stood out to us was that, one of the possibilities with the functional connectivity has always been, is it the vascular coupling? And I think that with some of the data that Patrick Pruitt here analyzed for us, looking at the ASL, there's no evidence that we're consistently increasing the cerebral blood flow, which then lets us move down the rabbit hole of saying, well, is it synaptogenesis? Is it increased white matter density? What is driving these effects? So in those 2, as Kayla pointed out, what was interesting to us was kind of the absence of any clear pattern of biomarker deviation. And so I think that has us scratching our heads a little bit. And I think one of the things that Michael Minfong and I were talking about yesterday was, do we need to look at medications? Are there other things that we should look at? And unfortunately, with those cases, I don't think we had flare sequences, because that was the other possibilities. Maybe there's something weird with white matter. So I think it opens up a lot of questions, not to steal your thunder here, so I'll hand it back over. But, I think the other thing that, as you saw with some of the previous slides, a lot of times folks are getting about 0.09 to somewhere around 0.25 volts per meter of delivered current. With these particular cases, we tried to get that up to about 1 volt per meter, because in the animal literature, and please correct me if I'm wrong, but I believe that the best dose response information from animal literature is there for above the equivalent of 1 volt per meter. And in most human studies, because we use traditional biases limit us with how much we're doing, we're in this much lower range. And so I think that, the results were not as robust as I had initially hoped, but we also, as you saw, tailored the stimulation montage to each individual's FDG. And now we've kind of circled back, and we're reevaluating that approach to take much more of a network-based. And so we're doing a lot with network-based stimulation, as I think Alex mentioned. So it's an iterative process. Hopefully we'll be able to kind of refine this. We did put a grant in for this. We'll be resubmitting that in the next cycle. So hopefully we'll be able to talk a lot more about some of the PCA work. Sorry, other questions for Kayla, not to hog the spotlight.
Mine's a quick one. Do you have any sense, so you have the 24-hour data, do you have any sense about the durability, potential durability of these effects? It's a neurodegenerative condition. I guess, any thoughts on that?
Yeah, she was asking about durability. So we did see some effects pre to post, and what about the durability of these effects? Did I get that correctly?
Yeah.
Okay. So we did do, again, if I'm understanding correctly, we did do 30 days post TDCS, or I'm sorry, 30 days post TDCS. We did another MRI and follow-up tasks as well. Now, I don't remember what the data looked like. I don't recall off the top of my head, but we did assess for that. Now, again, Dr. Hampton will probably know better.
So we've been mapping this out across a number of different studies. So we have a double-blind RCT that was VA-funded trial where we measured fMRI 3 to 4 days post the last session, which we did 5 consecutive daily sessions. We saw a beautiful physiologic change that persisted 3 to 4 days later. With some of the pilot studies that we've been doing, we've been kind of mapping this out, and we actually see better physiologic response 24 hours after the last session. So with some people, we evaluated them, I think like an hour after the last session, 24 hours later, 48 hours later. And that 24 hours seemed to be really nice in some of those case studies. So we've started to try and target that with a lot of the pilot work that we're doing, because I'm not sure that the immediate effects of stimulation map on to what we see later. Now, one of the big questions is the durability. I think that, again, just to get on a little bit of a soapbox, I think that neuromodulation is held to an unfair standard. We know that if we get a blood pressure med, it's going to take time to work. And if you discontinue it without changing diet or exercise, all things being held equal, your high blood pressure is going to come back if you discontinue that medication. So why is neuromodulation different? We're working with diseased brains that have been diseased for years. So do we think that single sessions or even brief bouts would be sufficient to enact long-term change? And I think that that's where the field, in my opinion, is unfairly treated. We've never asked that of a pharmacologic agent or other interventions. So I think that it's a fantastic question, but to me, I think that's also some of the work that we're doing with at-home stimulation and really trying to do personalized stimulation in home environments so that we can have that sustained effect. Now, how long, how much? No clue. Nobody knows. Great questions for the future. Sorry, I've gone on real quick, Voiko, super quick. So the question was, did we map visual cortices and all the different areas of the primary visual cortex equally susceptible? Fantastic question. The data are really cool with these PCA patients. Some of them have this beautiful island of preservation in primary occipital. And then as you get into the ventral and the dorsal visual stream, it's dorked. Others have primary occipital dysfunction, tau and FDG, and the rest of the cortex is relatively intact. So it's a fantastic question, Voiko. And one of the things that we are doing is we've started a collaboration that Ari is pushing forward through the center, through us, with some vision scientists that have a number of tasks that within about 15 minutes will systematically map these different areas. So fingers crossed that takes off as well. Fantastic question, though. So sorry, Kayla, I didn't mean to steal your thunder, but thank you so much for presenting. Great job.